Thank you, Lena. And your commentary on ADC conjugation was a beautiful segue to another audience question that I'm going to point towards Meinhard, because the question asker pointed it towards Meinhard. Krupali asks, Meinhard, how do you determine conjugation efficiency in process samples for each step during ADC conjugation?
Yeah. I think just a pre-word here. So we at SOTIO, we have established different conjugation technologies, so that most of them are enzymatic conjugation technologies. But nevertheless, we typically establish the conjugation efficiency based on the conversion of the molecular antibody into an intact ADC with the desired DAR value, and everything which is only partly conjugated or that has to be removed by the ADC downstream process. So this is how we establish, let's say, the efficiency. And this, as I mentioned, this should be quite high because it's a high cost unit operation, and therefore quite extensive DOE parameter studies are performed here. And the test itself, the DAR, this is a typical HPLC reverse phase test that can be applied here.
Excellent. Thank you, Meinhard. It's 23 minutes after 11 already. This is fantastic. You guys are doing a great job. Good audience questions coming in, and we'll get to some more of those here shortly. But Heather, I would like to ask you to pull up our first polling question for the audience. So this is for you, our guests. Polling question number 1. In your organization, are parameters like titer, PTMs, and product quality addressed primarily during, choose 1, cell line selection, cell clone selection, or during cell culture development? So take a minute to answer those questions, or that question. And while you're answering that question, I'm going to ask our panelists if they have any guesses or want to do any prognosticating on how this question will be answered. Lena, what's your take? What if you had to put money on A, B, or C? Which one would you place your dollar on?
Hopefully, I would put money on A.
Meinhard, would you concur?
I think we would include, I think, all three phases where we address PTMs. So cell line decision, I think it's the first, of course, but also during clone selection and then cell culture, you can still then make changes on the molecule, and then this is how I would see it here.
Yeah. Neeraj, I don't know if the implications of that question affect anything further downstream, but if you have an opinion here, you're welcome to weigh in as well.
I agree with Meinhard. I think of all 3 stages, I think you need it, but also during the production, I think from I would say a manufacturing consistency standpoint, you still need to measure the productivity, which is the titer, has a direct implication there, and the PTMs. Yeah, I think these are pretty critical parameters right from the get-go.
Yeah. All right, let's take a look at how our audience responded. During cell clone selection is the leader, but it's a pretty even split, so that kind of aligns with, maybe that aligns with Meinhard. They had to select one. They couldn't select all 3, so maybe we should have made that a select as many as apply option.